Next story. Unusual habit. Human hosts as frequent jumping grounds. In a groundbreaking study published in Nature Ecology and Evolution, scientists have challenged the prevailing notion that viral host jumps predominantly originate from animals and move to humans. The research, conducted by Tan et al., delves into the evolutionary mechanisms that underlie these zoonotic, animal-to-human, and anthroponotic, human-to-animal, transmissions. The team analyzed genetic evolution patterns in viruses associated with zoonotic and anthroponotic host jumps, using an extensive dataset of viral genomic data. Their findings reveal that humans are as much a source of viral spillover events to other animals as they are recipients. To address the challenge of non-uniform viral taxonomy, the researchers developed an automated protocol that constructed closely related, viral cliques, as operational taxonomic units. This grouped together different viral species into single cliques and, in other cases, split a single species into distinct cliques. The resulting 5,128 cliques served as a standardized framework for categorizing viruses on the basis of genetic similarity rather than relying on traditional, often inconsistent, taxonomy. The authors inferred phylogenies for all viral cliques and studied the changes that happened on these host jump branches. Although most viral cliques showed no jumps, some revealed frequent jumps to new hosts. Interestingly, viral host jumps from humans to animals tended to occur almost twice as frequently as the other way around. The study also found that certain viruses have advantageous mutations to infect new hosts and are thus predisposed to jumping between hosts. However, the authors acknowledge the limitations of their research, including the sampling bias inherent in the dataset and the need for more systematic and uniform data collection practices. The findings of this study provide interesting insights into the complex dynamics of viral host jumps but also underscore the need for more systematic genomic surveillance and a collaborative global health framework that transcends species and geographical boundaries. By integrating comprehensive viral genomic data with ecological and environmental insights, we might get better at predicting, preventing and mitigating viral spillovers. Source: Less than nature greater than. Next story. Unlocking the potential of copper for creating chiral amines. Next story. A breakthrough in asymmetric amination, a new method for creating complex molecules. A team of researchers, led by Feng Zhou, Xin Wang, and Jian Zhou, have developed a groundbreaking new method for creating complex molecules known as chiral alpha tertiary amines, ATA, using copper-catalyzed asymmetric amination, ACPA-1. This new method, reported in Nature Chemistry, allows for the creation of these important structural motifs in high enantiomer purities, or high levels of purity in terms of their three-dimensional structure. Chiral amines are a crucial class of molecules found in many natural products and pharmaceuticals, and the ability to create them in high levels of purity is a significant challenge in the field of synthetic chemistry. The new ACPA method uses sterically confined ligands to create ATAs with high levels of enantiomer selectivity, or the ability to create only one of the two possible mirror image forms of the molecule. The new ACPA method is a significant improvement over previous methods for creating ATAs, which often required the use of specific electrophile classes or the formation of ACN bond by stereoselective substitution at a tertiary carbon center. These methods can be complicated by the fact that ketamines, which are intermediate compounds in the formation of ATAs, can exist as E or Z stereoisomers, and separation of these isomers is not always straightforward. The new ACPA method, on the other hand, uses a dynamic kinetic asymmetric transformation for the deracemization of easily prepared tert-butyl carbonyloxy activated propygylic alcohols to propargyl ATAs. This means that the method can create the desired ATA molecules in high levels of purity, even when starting with a mixture of E and Z stereoisomers. Furthermore, the alkene functional group present in the propargyl ATAs created using the new ACPA method represents a powerful synthetic archetype that can be reacted in many different chemical processes. The researchers demonstrate this by converting the ACPA products to 5- and 6-membered heterocyclic ATAs, including the synthesis of a BACE1 inhibitor target, and enabling several diversity-oriented synthesis applications to complex heterocycles and diamines. Overall, the new ACPA method represents a significant advance in the field of synthetic chemistry and the creation of complex molecules. Its ability to create chiral amines in high levels of purity using sterically confined ligands is a major step forward in the field, and its potential for creating a wide variety of other complex molecules is vast. Source: Zhang, Z. al. Copper-catalyzed asymmetric amination. Nat. Chem. 2024. Less than DOI greater than. Next story. Navigating the intricate world of tryptophan interactions, the art of blocking and rescuing. Scientists have developed a new method to control and manipulate specific tryptophan residues in proteins, allowing for precise control over protein function. This new technology, published in Nature Chemistry, utilizes a vinyl-caged tryptophan analog that can be selectively decaged to rescue protein activity. The field of genetic code expansion, GCE, has rapidly advanced in recent years, with new technologies in codon reassignment, synthetic genomes, and evolved AARS, tRNA systems. The incorporation of non-canonical amino acids, NCAAs, into proteins allows for the manipulation of protein function by targeting specific residues within an active site. However, until now, this technology has primarily been applied to amino acid residues that contain polar side chains, such as lysine, serine, tyrosine, and histidine. In this new study, researchers have developed a method to cage the indole nitrogen of tryptophan with a vinyl handle, creating a vinyl tryptophan, VYW, that can be selectively decaged through the fast, specific, and bioorthogonal inverse electron demand Diels-Alder (IEDDA) reaction. This method allows for precise control over any protein that has a tryptophan residue that is critical to its normal function. This technology brings precision control that can enable researchers to better understand the potential importance of select tryptophan residues in proteins of interest, said the study's lead author, Dr. Chen. The researchers demonstrate the broad utility of their chemical activation of genetically encoded caged tryptophan system, termed TRP cage, in a variety of protein families and show that their optimized IEDDA-DIN reagent effectively rescues tryptophan interactions, thereby restoring protein functionality. This methodology has the potential to enable researchers to better understand the potential importance of select tryptophan residues in proteins of interest, said Dr. Chen. The researchers also developed a bespoke AARS for VYW and extensively tested the system in a wide variety of classes of proteins, demonstrating its high fidelity, high efficiency, and broad utility in establishing control over the activity of many proteins. In future works, this IEDDA decaging model might be applicable for studying other amino acids of catalytic or structural importance in proteins, said Dr. Chen. This new technology has the potential to have a significant impact on the field of protein research and could have numerous applications in the development of new drugs and therapies. Source: Zhu, Y. al. 2024, Blocking and Rescuing Tryptophan Interactions with a Vinyl-Caged Tryptophan Analog. Nature Chemistry. DOI 10.1038 per second 41557 024 01463 7. Next story. Unveiling the hidden volcanic threat of Santorini. Next story. The hidden eruptions of Santorini, a volcanic hazard reassessment. The pristine beaches and azure waters of Santorini, a Greek island in the Aegean Sea, belie the volatile past of this iconic volcanic caldera. A recent study by Preen et al. 2024 has unveiled a hidden chapter in Santorini's eruptive history, revealing that large explosive eruptions have occurred since the caldera formed, challenging the assumption that volcanic activity wanes after such events. Calderas, massive craters formed by large-scale volcanic explosions, are often associated with muted volcanic activity. However, the study of Santorini's caldera, which was created by the famous Minoan eruption, has revealed a different story. An international team of scientists aboard the research vessel JOIDES Resolution discovered a thick sequence of volcanic ash and pumice beneath the caldera floor, much of which is younger than the Minoan eruption, Preen et al. 2024. The team collected sediment cores and deployed geophysical instruments in the undersea basin of the caldera and the surrounding seafloor. Their analysis revealed that the basin contains both lava and substantial amounts of ash and pumice. High-resolution seismic reflection data and drill cores were used to correlate thick deposits of pumice and ash with thin ash deposits found within 40 kilometers, providing clear evidence of a large explosive eruption that occurred around 726 BC, only hundreds of years after the Minoan eruption, Preen et al. 2024. This VEI-5 eruption, comparable in size to the recent Hunga Tonga eruption, demonstrates that magma reservoirs can quickly refill with gas-rich magma. This finding challenges the traditional view that after caldera formation, the magma reservoir beneath only refills slowly and dispels the idea that eruptions soon after caldera formation are likely to be small and mildly explosive. The implications of this discovery extend beyond Santorini, as it highlights the potential for large explosive eruptions even soon after caldera-forming events. For instance, Taupo volcano in Aotearoa, New Zealand experienced a caldera-forming eruption 1,500 to 1,900 years ago and has recently experienced swarms of volcanic earthquakes that suggest magma is re-entering the reservoir beneath. The Santorini data suggests that reservoirs can refill relatively quickly, so large eruptions at Taupo could be possible even relatively soon after a caldera-forming event. The findings of Preen et al. 2024 underscore the importance of understanding past eruption history, in addition to present-day geophysical and geochemical volcano monitoring, to forecast the timing and magnitude of eruptions more accurately. While a large eruption within the next few years is extremely unlikely at either Santorini or Taupo, understanding worst-case eruption scenarios is crucial for hazard mitigation. The study by Preen et al. 2024 exemplifies the utility of integrating geophysics with sediment cores to unravel the eruptive history of submarine volcanic systems following a large caldera-forming event. Their findings serve as a reminder that large explosive eruptions are possible even soon after caldera-forming events and that current hazard models may have underestimated the potential of Santorini to produce large explosive eruptions. Source: Less Than Nature Greater Than. Next story. Mastering the STING pathway for enhanced immunotherapy. Scientists have developed a new approach to cancer immunotherapy by controlling the STING pathway, which could potentially increase the rate and durability of immunotherapy responses. The STING pathway is an evolutionarily ancient pattern recognition receptor that has been implicated as a master regulator of the cancer immunity cycle. However, clinical trials of STING agonists in cancer patients have yielded disappointing results due to major challenges in STING agonist design and delivery. To address this, a team of researchers has developed a genetically engineered, universal, STING agonist, unisting, that represents a fundamentally new approach. The unisting is designed to tetramerize outside of the endoplasmic reticulum, allowing it to recruit TBK1 and activate IRF3, but without engaging the NF-kappa B axis. This results in an innate immune response shaped primarily by IRF3 signaling, characterized by IFN-IS and interferon-stimulated genes with minimal induction of NF-kappa B-driven pro-inflammatory cytokines. The team has also packaged mRNA encoding unisting into a previously described lipid nanoparticle, LNP, formulation containing the bioreducible ionizable lipid coatsum SSOP. This formulation increases gene expression compared to LNPs fabricated using the conventional MC3 ionizable lipid and bias delivery to cancer cells over infiltrating immune cells. The authors primarily utilized an intratumoral administration route, demonstrating superior anti-tumor efficacy compared to CDN STING agonists in models of lung cancer, melanoma, and breast cancer. The unisting can be employed universally, as it can activate STING signaling in cells that otherwise lack functional STING protein. This is important because some cancer cells silence or down-regulate STING to evade the immune system. The recent advances in LNP design provide an opportunity to develop and optimize the nanocarrier for delivery of unisting mRNA to extrahepatic tumor tissue. The work by Wang and Li et al. Challenges us to think beyond conventional nanoscale design paradigms and more deeply into how to better control the cell biology that underlies responses to emerging therapeutics. As STING agonists continue to enter clinical evaluation, it will also be important to consider if potently activating the NF-kappa B axis is detrimental. The research was published in Nature Nanotechnology and is available at less than DOI greater than source. Wang, Y et al. Controlling the STING pathway to improve immunotherapy. Nat. Nanotechnol. DOI. 2024. Next story. Uncovering the distinctive topography of individual proteins and proteoforms. Scientists have developed a new method for sequencing and identifying proteins at the single molecule level, which could revolutionize the field of proteomics. The technique, called FRETX, uses a distance-based mapping strategy and single-molecule fluorescence resonance energy transfer (FRET) via DNA exchange, FretX, to create a full-length fingerprint of intact protein sequences. FretX works by attaching a DNA strand to the C-terminus of a protein, which can then transiently bind to a complementary sequence fused to the biomolecule of interest. A Fret donor and acceptor are then added, and the distance between them is measured using Fret. By optimizing the length of the DNA strands, the researchers were able to achieve a precision of delta E approximately equals 0.03 from 10 docking events. The researchers tested FretX on a range of protein samples, including disordered alpha-synuclein, ASN, globular proteins, protein biomarkers of bacterial and COVID-19 infections, and post-translationally modified ASN. They were able to accurately identify the position of single cysteines along the ASN protein, even when they were only 5 amino acids apart. They also demonstrated that FretX could detect post-translational modifications, PTMs, such as OGL canacylation. One of the key advantages of FretX is that it can analyze proteins from different sources, such as cell extracts and body fluids. The researchers propose a two-step bioconjugation reaction that enables the analysis of intact proteins with a solvent-exposed N-terminus group. They also demonstrate that pairing N and C terminal information increases the fingerprinting accuracy of ASN to greater than 85%. The ability to fingerprint protein sequences at the single molecule level is a significant leap forward for proteomics. While there is still work to be done to expand sequence coverage, integrate additional PTMs, and increase the distance range of observable residues to larger proteins, the development of FretX represents a major breakthrough in the field. The researchers believe that FretX and other similar techniques will soon transform protein identification into a reality, just as they did for nucleic acids. The ability to analyze proteins at the single molecule level will have numerous applications in fields such as medicine, biology, and biotechnology. Source: Less Than Nature Greater Than Next Story. Enhancing Single Molecule Transistors with Graphene Edge Interference. Graphene edge interference boosts single-molecule transistors. In a groundbreaking development for molecular electronics, researchers have harnessed the power of graphene edge interference to create highly efficient single-molecule transistors. This new technology, reported in Nature Nanotechnology, offers a promising solution to the challenges of fast switching speed, stability, and efficiency that have plagued previous attempts at molecular electronics. Molecular electronics, the ultimate downscaling limit of transistors, has long been a holy grail for researchers. The ability to control and understand molecules at the nanoscale interface has led to the demonstration of several single-molecule transistors. However, achieving fast switching speed, good stability, and efficiency has remained elusive. Now, a team of researchers has reported a single-molecule 3-terminal transistor that overcomes some of these limitations. The devices use graphene electrodes and a voltage gate, showing exceptional properties due to the harnessing of quantum interference effects. The researchers first created graphene electrodes with irregular wedge-shaped protrusions, where 2 zigzag edges merge into a locally armchair edge. This is near the presumed molecular binding site in the devices. They then attached a porphyrin molecule to the graphene wedges. The quantum nature of electrons traveling across molecular scale circuits gives rise to fascinating effects arising from the phase-coherent character of electron transport. Destructive quantum interference, DQI, is the suppression of electron transmission at certain energies arising from the phase shifts of different orbital resonances involved in transport or of different spatial pathways. In the devices reported, DQI appears in the graphene electrodes, leading to a suppression of electron transmission at certain energies. The researchers harnessed this DQI antiresonance to define the off state of the transistor, while energies close to molecular resonances, where transmission is high, define the ON state. By changing the gate voltage of the device, they could tune molecular levels between these two states, achieving an on-off ratio of 104. This value is 2 orders of magnitude higher than previous single-molecule transistors using O electrodes. The sharp DQI antiresonance allows for simultaneous high on-off ratios and efficient switching, with the devices exhibiting switching at the thermionic limit. The chip-like architecture allows for rapid modulation of the gate voltage and robust switching in the KHZ regime, much faster than the frequencies achieved by electrochemical gating in previous single-molecule transistors. The devices are also very stable, with the ability to switch for 105 cycles at different temperatures over several weeks. DQI in the graphene electrodes is robust, and the chemical bonds to the graphene edges are more stable than usual pi-pi stacking in molecule graphene interfaces. While the devices currently only work at temperatures up to 100 K, the potential for this technology is enormous. Atomic scale characterization of the graphene interface and progress in the creation of tailored graphene edges are immediate research directions that can be undertaken to address the outstanding issues of the devices presented by Chen et al. This work is an exciting step in single-molecule transistor development, demonstrating the potential of graphene edge interference to revolutionize molecular electronics. Source: less than nature greater than